All right, staying on time. Perfect. I'd like to introduce our last speaker before the break, which will be at 2:50 to 3:05. And that is Dr. Jimena Laporta from the University of Wisconsin. And her talk was titled In-utero heat stress impairs mammary gland development and future productivity. The floor is
Can you hear
yours.
me okay? I have to stay here. I should ask, can you see me? Because I'm not sure. So I'll just move these a little bit there. Okay. All right. Well, welcome. Good afternoon, everybody. I would like to thank the organizers for the invitation to speak today. I will try to keep up after Dr. Casey. It's a great honor to be here today. This is going to be the, I don't know where to look. Okay. This is going to be the outline of my presentation. We're going to be talking a little about the overview of mammary gland development. We're going to introduce the windows of developmental plasticity of the mammary gland, and then we'll delve into the short and long-term effects of in-utero heat stress on the progeny. And I'm sure a lot of you in this room will agree with me in that the mammary gland is a unique and fascinating organ at the same time. It's unique in the sense that most of its functional development will take place postnatally, unlike many other organs. And it's going to be exposed to hormones, the mammogenic hormones and lactogenic hormones, and also to environmental factors that are going to be influencing the developmental trajectory of the mammary gland. And it's unique in that it's one of the only organs that undergo these repeated cycles of functional differentiation, lactation, and then regression across many times across the lifetime of a cow. So one of the goals of the dairy industry in general is to generate heifers and cows with mammary glands that can produce high amounts of milk, of high quality milk. So the question becomes, first, how, and second, when do we do that throughout this developmental progression of the mammary gland to really generate an impactful and long-lasting effect? So if you took lactation or you teach lactation 101, you'll teach your students that milk output is some sort of a simplified function of the number of mammary epithelial cells that are present within that gland. And the other side of the equation would be how much milk each of those cells can synthesize, or in other words, their metabolic activity. And we were just talking about that with Ben and Christy over lunch, so it's really fun lunch, and they're students. So this concept of developmental plasticity or plasticity of the mammary tissue is not a new concept. In fact, I really like this review by Buttenard and collaborators, where they really showcase mammary plasticity during lactation when these cows are challenged, right? We challenge them voluntarily, such as when we impose milking frequencies, different milking frequencies, or photoperiod and stuff like that, that Dr. Casey was just talking about, but also involuntary challenges, like, for example, the climate, which is what I have been working mostly, heat stress. And the mammary gland is able to adapt, to respond by altering either the number of mammary epithelial cells that they have, or the activity, or both at the same time. And this response can be immediate or transient and go away after the challenge is gone, or it can be persistent and carry over long after the challenge is gone. But this plasticity of the mammary gland is not exclusive to lactation, and it's also during early life. And in this review, Dr. Akers, actually this paper is from last BOFA in 2017. He really showcased and he curated several experiments in different species showing that the tissue foundation that is created during early life, and we're going to define what that is later, can really affect future mammary gland development and function, possibly through better supporting the massive growth of the mammary gland that we see later on in gestation, in the puberty, gestation, and later stages. So what are these windows, early windows of mammary development? I like this review as well from Nate and Sorensen. They put these multi-species examples of neonatal mammary gland development abnormalities and the potential consequences that might have in adult life. A few examples to highlight here is excessive estrogen, exposure to excessive estrogens during early life can lead to precocious proliferation of the gland, and that can increase the risk of breast cancer. And I just saw Russ over there here, so you know all about that. Obesity, that's another big one, right? Leads to impaired mammary development and lactation failure. And they cite the fat heifer syndrome. And that simply means this peripubertal overfeeding leading to reduced milk yield later on in life. So traditionally, I should say historically, in bariatric nutritionists, for the most part, have tried to focus on prepubertal nutrition to improve parenchyma growth and hopefully, oh my goodness, and hopefully improve milk production. Sorry, I talk a lot with my hands, so I'm trying to keep quiet here. To improve parenchyma growth and future lactation. I think we have enough evidence now to show that this really impairs mammary gland development and it reduces milk yields, and that it really has a persistent effect through life. And more recent evidence has shown that pre-weaning nutrition, that early mammary gland is responsive to nutrients, and pre-weaning nutrition has been shown to enhance mammary growth, and this exerts positive effects in lactation. There's a handful of studies showing this, but I feel like this is a very interesting line of research, and it really illustrates how divergent this effect of this timing of feeding or the plane of nutrition and really showcase the plasticity of the mammary gland, how it can adapt. So we talk about peripubertal, pre-weaning mammary developmental phases, but what about the prenatal mammary development phase? We typically say, again, in our lactation 101, they're born with teeth well developed, and there's this rudimentary ductal system present at birth. But if you think about the mammary gland parenchyma, really that we observe during lactation, really arises from a single fetal epithelial bud of mesodermal origin that really shows very early in gestation. At about a month into gestation, you can see that bud arising. And from there are very highly regulated and dynamic processes that you all know more about than me, but cell migration, cell differentiation, the establishment of epithelial structures, cell lineage determination, that's very important during that time, and tissue crosstalk between those epithelial structures, the stroma, the fat pad, and stuff like that. So we know that the microenvironment in which all these processes take place, those systemic and local factors, and having the right signals at the right time is very important for these to occur normally, right? So I feel that sometimes this developmental phase is neglected in a way. And I think there's a lot more going on there than we think. This is just showcasing fetal mammary gland. I think it's at the 100 days in gestational age. And we see those epithelial cells and canalization going on. We see myoepithelial cells, and we see a lot of proliferation. I think they show here 20% increase in proliferation there. And that is leading to this rudimentary tree, which we can see those primary, secondary, tertiary bags infiltrating that fat pad. Now, what I think, and others think as well, this is not me making this up, that this early ductal growth that is established in utero is really crucial for providing what we say the scaffolding for future lobular development during puberty with those ester cycles and really during gestation where multiple mammary gland growth will occur, and that will lead to a successful lactation. Now, what we do or we're trying to understand is how this, because this occurs in utero, it's subject to fetal programming, such as every other organ in the body. So how this suboptimal intrauterine environment, specifically hyperthermia, intrauterine hyperthermia, how that might derail the developmental trajectory of the mammary gland during this stage, and how that can have an impact later in life. So this takes me to the model that we use, which is the dry pregnant cow model. And I'm sure most of you here know what a dry cow is, but pretty much is non-lactating period between two lactations. And it's a unique model because it allowed us to study the mammary gland development of the dam, but also because it coincides or overlaps with late gestation where that fetus is growing exponentially, we can study fetal programming as well of every organ, but we focus mostly on the mammary gland. And so we know that during that period, there's a lot of cellular processes occurring that are greatly important for proper involution and redevelopment as parturition approaches. And if we mess with those, that can have consequences in the next lactation of the cow. And when I started this line of research about 5-6 years ago, I can remember, in collaboration with Dr. Dahl at the University of Florida, we knew a thing or two about the dry cow at that time. And so we knew that dry period heat stress really affects the involution process of the mammary gland, and that leads to reductions in milk production of about 5 kilos less per day. So really that insult during the dry period impacts future lactation of the dam. But we knew also that intrauterine hyperthermia was impacting the growth and survival of the offspring. And a retrospective study show that milk yield might be also impacted of that offspring. And I see, okay, I can use that. So what I wanted to do as a curious lactation biologist, as Teresa said, we like asking questions. So I started asking questions, how, why, when these things were happening. So we used this model where we either heat stress the pregnant cow during the entire dry period, or we offer heat abatement to that cow to sort of take her back to thermal neutrality, and then I follow the progeny. So we started this series of experiments that we're going to talk about today. So I'm going to briefly show you the short and long-term impacts of this in utero heat stress. We're going to go and talk about the mammary histology and the methylation landscape of these tissues during the first lactation of those daughters. And we're going to go back. I did it backwards. That's the story of my life. So then we went back and looked at earlier stages of development, mammary gland development. So we're going to go through these, and we're going to start with the short and long-term effects. So there's a host of effects in the offspring. We talk about some of these already with intrauterine growth restriction, and not surprisingly, they are very similar. One of the hallmarks of in utero heat stress is a reduction in gestation length. On average, we see 4 to 5 days reduction in gestation length. Reduced body weight, birth body weight of about 5 to 8 kilos at birth, and that impairment in growth is maintained through weaning, so that difference stays there for 60 days. And also the average daily gain. These animals gain less weight throughout that pre-weaning period. In one of the latest projects that we did, I wanted to know if it was just body weight or if it was more body size. So we did a lot of measurements in those calves at birth and throughout weaning. And we generally see that those animals are really smaller. They have shorter hips, height, body length is reduced, the chest girth is reduced, and surprisingly, the head circumference is really small. So it's really intrauterine growth restriction going on. I did not mention that we impose these treatments for 46 days. So it's, it's not a long time, but we still are dealing with this chronic heat stress. Some of the hallmarks of in-utero heat stress: innate immunity. We see the poor efficiency of immunoglobulin absorption of those calves is greatly impaired, and these immunoglobulins are lower in the circulation for the entire pre-weaning period. So really they are having a hard time absorbing immunoglobulin. So there's something going on in utero in the gut that it's really messing with that. Adaptive immunity, we look at many things and it's impaired as well. Grain intake, they eat less, especially between week 5 and 8, where you really want them to pick up with grain intake, they really don't. Thermoregulation, we see these animals coming from in utero heat stress have core body temperatures that are higher. I believe it's 0.2 degrees Celsius higher than the ones that were in utero cool. They are trying to thermoregulate, they sweat more, so they're really messed up there. And we see also what I would call medium to long-term effects. Age of puberty attainment is decreasing those animals. And I put this as an example here, heat height. We can see that in red, we have the in utero heat stress daughters or heifers. And we see that even up to 300 days of age, and we just took the 360 days a few weeks ago measurement, they're still shorter. So this is for you, Dr. Yates, that you were saying, oh, we keep them until 60, well, we keep them until 360 days, and we're still collecting measurements. So thank you to my students that actually do all the work. So just showcasing that these are really long-term effects that no matter where they are, because these heifers were born in Florida, I took them to Wisconsin, they went through summer, winter, and you still see this difference. So there's something really bad going on in you when those dams are exposed to hypothermia during just 46 days. So to showcase the long-term effects of in utero heat stress, we did this kind of in-house meta-analysis where we put together 10 years worth of data from the University of Florida, where we had 200 dams that were heat stress and 200 dams, polar dams, multiple scouts that were provided with heat abatement during the dry period. We have about 70 daughters per group that were born to these dams, and we were able to collect data from 25 granddaughters in each group. So with that data set in hand, we could ask these long-term questions. What about their productive life? What about their lifespan? What about that milk yield reductions that we see? Is this just a one-time thing, or is a multi-lactational effect? What about multiple generations, those granddaughters that I was telling you that are born to the daughters that were born to a cow that was under heat stress? So a mouthful, but that's what we're trying to do here. So this is the survival analysis of the daughters coming from a heat stress dam in red, in the red line, or daughters coming from a cool dam. And here we have the age on the x-axis, and we have different life events in here. Women, first insemination, calving, 3, 4, 5 years of age. And you can see a clear pattern here of that red line, again representing those heat stress daughters, but it's always below the black line. Okay, you see all the way through. So in utero heat stress reduces survival to first calving, so less heifers are making it to calving, to lactation actually. And when you look, those that make it to lactation, the survival is reduced through third, fourth, and fifth year, which corresponds to second, third, and fourth lactation. And overall, productive life is reduced by 5 months, and lifespan up to 1 year. So it's really impactful effects that we see in the daughters of these dams. So those that actually make it to the first lactation, this is milk production throughout 35 weeks in milk. We see the same trend, that red line below that black line throughout the 35 weeks in milk. They start producing less and they continue to produce less throughout those 35 weeks in milk. Overall, they produce 2.2 kilos less per day. So really impactful as well. When we look at their second lactation of those that make it to a second lactation, we see the same trend. That red line again below that blue line, these, remember, are the daughters that were born to dams that were either heat stress or cool or provided heat abatement. They produce 2.3 kilos less per day throughout 35 weeks. And those that make it to the third lactation, which are actually not that many at this point, you see again that same trend. We start to have a lot of variation here because we're dealing with 20 cows here. But I was surprised to see this, and it's really interesting. So we see reductions in milk, fat, and protein yields. I'm just showing the milk here just for the sake of time, but we see this for at least 3 lactations. So there's some sort of in-utero programming of this less productive phenotype that have very lifelong consequences on production and survival. So what are the granddaughters? Is this an impact on just one generation? We know that it impacts the dam, next lactation. I show you how it impacts the daughter, multiple lactations, but what about the granddaughters? This is the survival analysis of those granddaughters. Again, they were born to the daughters, right? So you can see the same trend here. That red line is below the black line. 25% less of those make it to breeding. Actually, 26% don't make it to breeding, and 27% of those don't make it to the 3rd year of life. So pretty big impacts there. And when we look at the first lactation, the milk production of those granddaughters, you see that they produce 1.3 kilos less milk throughout the lactation. So this late gestation heat stress has carryover effects that stay for at least 2 generations. We do not know what happens with the 3rd generation, but hopefully we can look into that soon. Now, we started to look at the mechanisms, why this is going on, and we went in, these are the daughters, that F1, we went in when they start lactating. So this is 2 years after they were born. They start lactating, and we went in and we took mammary biopsies at 21 and 42 days in milk. Again, of this in-utero heat stress or in-utero cool heifers. This is the first lactation. And actually, this work was done by Amy Skivel, who's back there. So if you have any questions, just direct them to her. So she did all this amazing work. So what we look into here was, I wanted to know the tissue microstructure. What was leading to that decrease in milk production that we see? So we did some staining, to basic staining, and we see this is the in-utero cool mammary glands, and these are the in-utero heat mammary glands. So if you have your glasses on, you can see that the difference is pretty obvious. You have the very nice round alveoli here with that lumen, very typical of a lactating cow at 3 weeks in milk. Whereas in the in-utero heat stress, you have this significantly reduced luminal alveolar area in those tissues. We also see higher proportion of connective, or that stroma tissue, which is really important. I'm not saying it's not, but during lactation, you want to maximize the secretory tissue for milk production. So the next question was, okay, are these smaller alveoli, the same number of alveoli, but are these smaller alveoli leading to less mammary epithelial cells capable of producing milk? So what Amy did, she went in and counted, trace every single alveoli and count every individual mammary epithelial cell there. We did some correlations, and we found that these smaller alveoli had smaller, less mammary epithelial cells capable of producing milk. We also saw that they have lower proliferation, so they were not only less cells, but they also were proliferating less. So this is my little model here. These are, to me, to us, undesirable characteristics during lactation. They have compromised synthetic and milk storage capacity. They have smaller alveoli with less mammary epithelial cells and less proliferating cells. So we think this is at least one of the contributing factors to this reduced lactation performance that we see. And this is just the first lactation of those heifers, but it's 2 years after they were in utero heat stress. So it's pretty impactful. Now, obviously, trying to keep opening dark boxes, I wanted to know why is this happening? How is this happening mechanistically? Where are the biological links between intrauterine conditions to the phenotypes that we're seeing later in life? And we decided to chase the ghost of the epigenome. We started looking at DNA methylation, just because it's one of the most stable and the epigenetic change that we know a little bit more about. And we thought that intrauterine hyperthermia might be triggering some of these epigenetic changes in utero, and they were sustained throughout the first lactation. So we started looking at that in this study. Again, Dr. Skibo here, she'll address your questions. We find 135 differentially methylated genes between in utero heat stress and in utero cool daughters. And we're going to go one by one with all this one. I'm not going to talk about all those genes, but I just highlighted a few here that were hypermethylated in utero heat stress heifers. PTK2 involved in cell migration and cell survival. Actually, I found a paper where a knockout mice model, but they showed that they were unable to lactate because those mammary glands were just undeveloped. WRN involving DNA repair. This gene PRKG1 involved in apoptotic induction and growth inhibition. And some genes that were hypomethylated that were interesting were SAP1 involved in the regulation of stem and progenitor cells, which is an area that it's very interesting to me, and I would like to look more into, but it was nice to find this gene that was involved there. LAP, an antimicrobial peptide, DTX2, involved in cell communication and in the notch signaling pathway, which some of you know that it's very, very conserved signaling pathway involved in development. So the next question was, okay, are these changes that we see in methylation, are these unique to a mammary gland under in utero heat stress? Or are there common patterns of DNA methylation that are induced by heat stress that are, regardless of sex, age, and tissue type? So we try to find a very different tissue to look at. We look at the liver of bull calves at birth. So anything more different to a lactating cow. But these livers were coming from bull calves that were also in utero heat stress or cooling. So we wanted to know if the differences that we were seeing were exclusive to the mammary gland. So we were happy to see that there were 50 genes actually that were shared between these two different organs and different timings and different genders. So we see these 50 genes that we're looking more into, but there are some interesting ones that we're really trying to chase. So we see some involvement in protein synthesis machinery. So as you know, this is very important for protein synthesis. If you're impacting the machinery that it's making proteins, you can have an impact in more proteins. And we see that those phenotypes. Mitochondrial function, we talk a lot about that during this meeting, particularly this gene IMMPL2. So energy for growth, energy for lactation, very important both for that. Cell hydration and migration, this PTK2 that I mentioned. Cell signaling, we see this phospholipase C that I'm sure all of you at some point in your career have seen it. So very fundamental processes that are involved in survival DNA. repair, very fundamental processes. So we're going to be looking more into these, and so stay tuned. All right, so we're going to go back to the last stages that we were looking at, the birth and weaning developmental phases. So remember, we see that after intrauterine hypothermia, we see that they produce less milk, right, 2 years after. So we see microstructural differences. We see a different methylation landscape in those glands when we compare them to those that were born to in utero cool dams. But we wanted to look back and look at the mammary developmental trajectory earlier in life, and we focus on these stages: birth, weaning, puberty, and gestation. I'm going to show you these two today, and we're currently working on puberty. These heifers that are being bred at the moment, they are 13 months of age now, and so we're taking mammary biopsies on those to look at these. But when we did, this is kind of our experimental design. We have cows that were, 40 cows that were heat stressed for 60 days during late gestation, and 40 cows that were provided heat abatement. And once those heifers were born, and they were bred to sex semen, so we had a lot of heifers here. So once they were born, they were all put together in individual hutches, and they were all raised for 56 days. And we did 2 euthanasias here. We euthanized a subset at birth and a subset at weaning. Day 0 and day 63, 1 week after weaning. So what we did is we dissected the mammary gland of these heifers, and you can tell, we did this in Florida, you can tell that by my hair here, all curly, very humid. And so we dissected the mammary gland. This is from the utero cool heifers. This is in utero heat stress heifers. And so what we did is we section the teat, we paraffin in bed, and we did H&E and look into the microstructure of that tissue. And then we peel off the parenchyma tissue. I don't know how well you can see there, but it's very small, but it's very clear, and you can actually peel it off from the fat pad. So we did that, and we reserved that for molecular work that I'm not going to present today, so I'll show you just the histological work. So at the we did the same. This time the mammary gland is a little larger, obviously. So we couldn't embed the entire teat, but what we did is we dissected from the fat pad, and we sectioned a small piece from the proximal parenchyma here, and we did the same process. We did H&E staining, and we did what these call our stitch images, where you have 200 4x images, and then the program put those all together. So it's kind of trying to mimic the whole mount of mouse mammary gland. So those that work with this understand what I'm talking about. It's very challenging to do all this in cows, but you can see here, this is Bethany, my PhD student. She's dissecting the parenchyma. Here you can clearly see the parenchyma and the fat pad. So what we found at birth, the mammary glands of in utero heat stress heifers, this is right at birth, 4 hours after birth, no feeding, no nothing. So they were just born to those dams. So we see those mammary glands, the trim mammary gland that you can see here, they weigh less. They have reduced fat pad and reduced parenchyma mass. And I don't know how well you can see this, but we see a 50% reduction in the parenchyma weight of those heifers, of those mammary glands. Then we look at different measurements of length measurements. We see they are shorter in length, and I don't know if you can see that from these figures. I tried to find different ones, but I don't know how well you can tell, so you have to bear with me here. They were shorter in length, and the distance between the front and the rear teats, you can see right here, that's reduced, and the distance between the front teats is reduced. So you can see how quite different they are. At the rear, we see the mammary glands of in utero heat stress heifers, again, they're smaller. So after 60 days of all of those heifers being raised together, no management differences, all the same. We still see differences in the weight of those mammary glands, reduced fat pad again. We don't see any more differences in parenchyma, so the parenchyma tissue seems to catch up, but not the fat pad and not the whole mammary gland. They are still shorter, but we don't see any more differences in the other measurements that we did. And looking into the more interesting stuff here, we did H&E staining as I showed you. And here we have the in-utero cool. This is day 0, so this is at birth. To look at the microstructure and the ductal epithelial structures at birth. And as they say, a picture is worth more than 1,000 words. So you can see how different developmental-wise they are. You see, we classify in these luminal and non-luminal ductal epithelial structures. So we can't count all those. And we see that not only the number of luminal, non-luminal, and obviously the total, but also the area of those structures was reduced in the uterine heat stress daughters' mammary glands at birth. So you see a reduction in both, and obviously in the total as well. At 63 days, so it's 1 week after weaning. Again, you can really tell from these two images here how they are different. We see a reduction in the area for the luminal and non-luminal. We don't see more differences in the count, so they have the same number of epithelial structures, ductal structures, but they are all just smaller. The last, proliferation in uterine heat stress heifers have decreased cellular proliferation in both compartments, the epithelium and the stroma. And we see that reduction here in the total. So we trace that and we counted the individual proliferating cells. And at weaning, the same, we see this 50% reduction in the proliferation rate of those compartments. When we look at the mammary fat pad, we see that in uterine heat stress daughters have more adipocytes, but those adipocytes were smaller, but we don't see that difference at the weaning. So in summary, in uterine heat stress, I hope I convinced you or I show you that it derails mammary development. There are fewer and less proliferative epithelial cells, epithelial structures, both at birth and at weaning, and this seems to persist into lactation. In uterine heat stress, epigenetically programs the mammary gland. There's this organ-specific methylation in the mammary gland, but there's also some differentially methylated genes that are shared between other tissues and other stages. So possibly this is true for other tissues. And this really leads to a less resilient or less productive cow for multiple lactations, at least 3 and at least 2 generations. So this is this phenotype persists for 2 generations. Is this transgenerational? We don't know yet because we have to look at that F3 that is not exposed to the insult, so we don't know that yet. So overall, long-term consequences of mammary development, compromising milk production later in life. Some of the work we're doing now, we're looking at that pubertal phase, looking at mammogenic hormones and tissue microstructure. We're going to move on into gestation, and we're going to look at the methylation landscape across all these stages. And we're generating the F2s, the granddaughters, to start looking more at the growth and molecular data, because all we have until now is some retrospective data on their survival and reproduction. So we're going to try to get more information on molecular information. So with that, I would like to thank Amy Skibo. Right here, she's now at the University of Idaho in the faculty. Jeff Dow for his collaboration at the University of Florida. Bethany Lato-Sen, she's my graduate student who did all this latest work. And these two are the ones that did most of the work I presented today, but really I have to thank all my current and past graduate students, and I need to update this picture. But they all, these experiments are long and tedious, and there's a lot going on. So everybody has to jump in, even if they're working on different things. So, and funding, obviously, that it's what makes this happen. So with that, I would like to thank you, and I'll take any questions.